Carbohydrate, structure and function. The purpose of this video is to look at the structure of monosaccharides, disaccharides and polysaccharides and relate those to the functions in living things. So first of all, looking at the word carbohydrate, that literally means made of carbon, hydrogen and oxygen. All carbohydrates have the general formula CH2O to the N. In other words, there are twice as many hydrogens as there are carbons and oxygens. The monosaccharide that we spend most of our time looking at is this one, which is glucose. Glucose is known as a hexose sugar because it contains 1, 2, 3, 4, 5, 6 carbon molecules. Now they are numbered clockwise from the oxygen, so carbon 1, 2, 3, 4, 5 and 6. The most significant ones are carbon 1 and carbon 4, as those are the carbons that contain the groups which will then be involved in bonding when we start to look at disaccharides and polysaccharides. Now, glucose is also able to show what we call isomerization. This particular diagram shows alpha glucose, but there is also beta glucose, which is shown in this diagram here. You will notice that the difference occurs here with specifically the OH group on carbon number 1. In alpha glucose, the OH group in carbon number 1 is below the plane of the ring, and in beta glucose it is above the plane of the ring. Perhaps the easiest way to remember this is ABBA, alpha below, beta above. Monosaccharide units need to be able to join to each other, and they do this through the formation of what is called a glycosidic bond. As you can see in this diagram, when they pair up next to each other, the carbon 1 on the left-hand unit and the carbon 4 on the right-hand unit come together, and the bond forms across the OH groups. So the bond forms across the OH groups with the elimination of water, and you form what's called the glycosidic bond here, which is across the remaining oxygen atom. This is called a 1,4 glycosidic bond because it is between carbon number 1 and carbon number 4. Technically it is an alpha 1,4 bond because it is between two alpha glucose units. This type of reaction is called a condensation reaction. And so what is happening in a condensation reaction is that you are joining together across the O and the H with the elimination of water. So you lose the water molecule. That is the condensation reaction. A hydrolysis reaction breaks the bond with the addition of the water molecule. Two monosaccharides will join together to make a disaccharide, and there are 3 that you need to be aware of. Sucrose, which is made up from glucose and fructose, maltose, which is glucose plus glucose, and lactose, which is galactose and glucose. Polysaccharides have 2 functions. They're either storage or for structure. In this section of the syllabus, you need to only look at the storage polysaccharides. In plants, starch is the storage compound, and in animals, glycogen is the storage compound. Starch itself is made up of 2 molecules called amylose and amylopectin. Now, regardless of whether it's in a plant or in an animal, all storage compounds have the same requirements. They have to be compact so that you can fit a lot of glucose units into a small space. They have to be insoluble so they don't affect the osmotic properties of the cell, and it has to be easy for them to hydrolyze or condense, so release glucose or take up more glucose. So All of the storage compounds that we're going to look at today are polymers of alpha glucose. So they all look a little bit like this, with alpha 1,4 glycosidic bonds joining the individual glucose units. Amylose, which is part of starch, is a straight chain alpha glucose polymer. Because it's a straight chain, it ends up forming a helix here, and the interesting thing about this helix is it is exactly the same diameter as an iodine molecule, and that is why iodine is the test for starch. But the key points to remember: alpha glucose polymer, alpha 1,4 glycosidic bonds, straight chain, makes a helix. Amylopectin, which is the other molecule in starch, is a branched chain. And so what you have is you have your main chain here, which is bonded by alpha 1,4 links, and then you will have an alpha 1,6 link here, which will attach a branch. So you can see here that you have a branched chain structure, and amylopectin will branch every 24 to 30 glucose units. Now what this does is it increases the number of available ends, if you like, for hydrolysis and condensation to occur over, making it more efficient to sort of take up glucose or to release it. This is just another way of looking at it. So you can see here we have the amylose straight chain, whereas here we have the amylopectin, which is branched, with the alpha 1,6 bond forming the branch. Glycogen has an almost identical structure to amylopectin, except that it is more highly branched, being branched every 8 to 12 glucose units. It is more highly branched because animals have a higher energy requirement than plants do, and that is why glycogen is more highly branched.